Hi, now I am going to discuss about gene chromosomes. The gene chromosomes were first found in the cells of salivary glands, guts, trachea, and some other parts of the diphtheria insects by E.G. Balbioni in 1881. There are two main types of gene chromosomes, that is polytene chromosome and Lambrecht chromosome. Now we will see about the polytene chromosome. The polytene chromosome is also known as salivary gland chromosomes because it is isolated in the salivary glands of these diphtheria insects. And they are also called polytene by Koller because of the presence of many chromonomata in this chromosome. These chromosomes are very large due to the presence of high DNA content, and it consists of a bundles of chromosomal fibrils. The process of reduplication of the strands without their separation is called endoduplications. The homologous polytene chromosomes always remains closely paired in the mitotic prophase. This is called the somatic pairing, and these chromosomes remain in the permanent prophase stage. The polytene chromosomes bear along their entire length a series of alternative dark bands and the light bands, or it is also called the interbands. The dark bands are narrow and broad disc-shaped structures. The dark bands are the heterochromatic regions and contains large amount of DNA, but they are genetically inactive. They are fuel-gen positive. For fuel-gen staining, they get color. And the light bands or interbands are fibrillar. They are fuel-gen negative. For fuel-gen staining, they get a pale color. They are color. They looking like colorless. They are euchromatin in nature and contains a small amount of DNA. Here this figure shows the balbionic ring, that is the puff-like structure is called the balbionic ring. And here the dot bands altered with the light bands. The light bands are called the interbands. Number, distribution and localization of the disc or the bands, that is the dark band and the light bands, are notably similar in homologous polytene chromosomes. A single band or adjacent band of polytene chromosomes produce local reversible swellings at certain stages of the development. These are called the chromosomal puffs or bulbs. We have already seen in the last diagram. The chromonomata of the polytenic chromosomes give out many series of loops laterally, and these loops or the rings are known as the Balbioni ring. They are rich in DNA and RNA. This is an example for the polytene chromosome. It will clearly explain what are the structures found in these chromosomes. This puff-like structure is the Balbioni ring. The dot bands, which is altered with the light bands, these are called the interbands. Here the chromonomata is present, and this bulb is together called the Balbioni ring. Next is the Lambrecht chromosome. The Lambrecht chromosomes are found in the nuclei of oocytes of many vertebrates like fishes, amphibians, reptiles, and birds. It is first discovered in the oocytes of salamander. The Lambrecht chromosome was first observed by Fleming in 1882, and the name was given by Ruckert in 1892. It is named as Lambrecht because it resembles like the brush which is used for cleaning the lamp. The Lambrecht chromosomes are also found in the spermatocytes of many animals and also found in the giant nucleus of an algae called Acetobacteria. They are present as a bivalent forms with the 4 chromatids. And these chromosomes are sometimes larger than even some larger than the polytene chromosomes of the salivary glands of the diphtherians. The chromosomal axis is formed by the highly condensed chromatin, and the lateral loops extend from the chromomeres. These lateral loops of the DNA are always symmetrical and formed due to the intense RNA synthesis. There are about 10,000 loops are present in the oocytes of salamander in a haploid set of chromosomes. but the lateral loops are absent in the centromere region of the chromosome. The loops of the Lamprecht chromosomes and the Balbioni rings of the polytene chromosomes are the sites of active genes. Here this figure shows the Lamprecht chromosome. This is the Lamprecht chromosome. Here the Lamprecht chromosome is enlarged form of Lamprecht chromosome. A small section is here, and a single loop is presented here. This is the chromatin loop. These are the chromatid. It is two sister chromatids are the chromatids. Here the centromere is there. Thank you.